Next, we're going to be looking at what's happening to the sarcomere during the cross-bridge cycle. So what's happening is the Z-discs are going to get closer together. The thin actin filaments are going to slide towards the middle of the sarcomere, or centrally. The I-band is going to shorten, and the H-zones are going to disappear. That also means the contiguous A-bands move closer. So you can see the relaxed sarcomere at the top image, and the contracted sarcomere down at the bottom. Let's look at another view, though, of what it looks like. All right, here we have on the left a sarcomere at rest, and on the right, a sarcomere that has contracted. Keep in mind that sarcomeres make up myofibrils. Sarcomeres are going to be repeated as units in a myofibril. So if every sarcomere shortens because of increased overlap between actin and myosin, the entire myofibril is going to shorten. And in a muscle cell, when all the myofibrils shorten, the muscle cell shortens. And in a muscle, when the muscle cells shorten, the organ, the skeletal muscle, shortens. So when we talk about skeletal muscle contraction, we talk about these three steps: neuromuscular junction, excitation, contraction, coupling, and cross-bridge cycling. But we can't forget the role of calcium as the final trigger for contraction. Calcium release, or an increase of calcium in the intracellular space of a skeletal muscle fiber, is what initiates the cross-bridge activity and the sliding of the filaments. The cross-bridge activity actually ends when calcium is actively pumped back into the sarcoplasmic reticulum. So now that we have all the steps for how does the skeletal muscle contract, we want to look a little closer at some of that terminology that I was vague about. You may have picked up on it, things like local depolarization, end-plate potential, membrane shift, cations, things like that. Well, that actually has an entire physiology behind it. Getting the action potential from the motor neuron to the skeletal muscle fiber is just a little bit more complicated, especially when it comes to what's happening at the junctional folds of the sarcolemma. So keep in mind, for a skeletal muscle fiber, there's usually sodium ions in greater abundance outside the cell. And there's usually potassium ions in greater abundance inside the cell. And both sodium and potassium are considered positive ions, also known as cations. And like plasma membranes of all the cells in the body, they all have a at-rest stage. When we're looking at skeletal muscle cells, we're looking at their at-rest stage kind of being the beginning and end of this action potential, or this electrical current that is generated inside the sarcolemma. And all cells have this kind of polarized state, where they have more of one type of ion outside and more of another type of ion inside. It's just seen in more contrast in both neurons and muscle fibers. But this voltage difference created because of charged ions being located in different places across the membrane is going to be useful for us generating electrical impulse, because we can move these ions as long as we open ion channels. There's going to be multiple steps when it comes to generating an action potential in a skeletal muscle fiber. The first step is going to be generation of an end plate potential. That's the local depolarization, or the local voltage change. Then we actually get depolarization, where we see the travel of that electrical current or action potential along the entire sarcolemma. And then step 3 is the reset stage, repolarization, where we restore the sarcolemma to its initial polarized state. So what does that look like? Well, it looks like an axon terminal that contains acetylcholine in its synaptic vesicles, releasing that acetylcholine because of an action potential, and that acetylcholine going across the synaptic cleft and binding to acetylcholine receptors in the plasma membrane of the skeletal muscle cell. So that's what we're seeing at step 1. When acetylcholine in green binds to the purple acetylcholine receptor, it's going to open up that channel, because that receptor is really an ion channel. That's going to allow a huge amount of sodium to enter the cell, and some potassium is going to exit, but we get overly a large amount of sodium entering the cell. Well, generally the inside of the plasma membrane for many cells, but especially skeletal muscle cells, is negative compared to the outside of the cell. And now we're suddenly opening up this ion channel, and we're allowing a ton of sodium that is positively charged to enter the cell. That's going to make the inside more positive compared to the negative it used to be. So that's going to depolarize the cell membrane on the inside away from the negative pole. That's where the term depolarization is coming from. And as we depolarize, that signal gets to travel away from just these junctional folds or local depolarization to other parts of the plasma membrane. And the other parts of the plasma membrane are going to contain separate sodium and potassium channels. And to further this depolarization, we're going to see sodium channels open and allow more influx of sodium inside the skeletal muscle cell. And we'll only see the potassium channels open when we need to reset the membrane, because we'll lose that positive and reset the inside of the membrane back to negative when we use the potassium channels, because potassium will exit instead of enter. There's a good video that sometimes is still present on YouTube called A&P Flicks Neurophysiology Part 2, Generating an Action Potential, that goes into the steps of how skeletal muscle uses an action potential. You can also look this up in a neuron, and a lot of the videos you're going to find on YouTube if you're looking for generation of an action potential are going to show you these steps in a neuron, but they are very similar in a skeletal muscle fiber. So let's go through the steps. So the process begins when the nerve impulse we call an action potential reaches the axon terminal, and it opens voltage-gated calcium channels in the axon terminal. Calcium going into the axon terminal causes the release of acetylcholine via exocytosis into the space called the synaptic cleft. So again, that's what we're seeing at step 1. That's going to lead to a local depolarization called a local voltage change, because the released acetylcholine will bind to acetylcholine receptors on the plasma membrane of a muscle cell called a sarcolemma, and that will open chemically gated channels. These are the same channels we call acetylcholine receptors, but when they're open, they allow both sodium and potassium cations to go through. And there's actually a greater influx or movement of sodium ions into the cell than potassium out of the cell. That causes the negative inside of a muscle cell to turn more positive or depolarize, and that creates a local voltage change or end plate potential. That local depolarization is the generation of the action potential in the sarcolemma. That local depolarization then opens downstream or further voltage-gated sodium channels that are in the next part of the sarcolemma. That's the beginning of not only generating, but allowing for travel or propagation of the action potential we've now got in the muscle cell. So that wave of depolarization travels to other segments of the sarcolemma, opening those sodium voltage-gated channels, allowing for further influx of sodium, further depolarization, and we'll move away from just kind of that local depolarization to a true depolarization, which is one of the first steps after reaching threshold in an action potential. And the transmission of that action potential along the sarcolemma will get to the T-tubules. Again, the sarcolemma and T-tubules are connected, and that will allow for the influx of the action potential inside of the cell and the opening of calcium channels of the sarcoplasmic reticulum that contain calcium, which abut the T-tubules. When we stimulate the sarcoplasmic reticulum calcium channels to release their calcium into the sarcoplasm or the cytosol of a skeletal muscle cell, that's going to increase the intracellular amount of calcium in the muscle cell, and it's the rise in sarcoplasmic calcium that triggers the removal of the tropomyosin troponin cascade and allows for the cross-bridge cycle or step 3 to occur. This would occur in very rapid succession. It's a very quick process, even though it's taking a while to talk about. So again, we get acetylcholine release from the axon terminal. It binds to acetylcholine receptor, which is an ion channel that opens. We see a big influx of sodium, even though some potassium leaves, and that depolarizes the skeletal muscle cell membrane. That depolarization can travel to further segments of the sarcolemma, further depolarizing it as it opens voltage-gated sodium channels, and this continues as the action potential, the electrical impulse, travels to the T-tubule, communicates with the sarcoplasmic reticulum, leads to the release of calcium, and then leads to step 3, the cross-bridge cycle. To reset, we would see the closing of the sodium voltage-gated channels and the opening of the potassium voltage-gated channels. And we would see a large efflux, or exiting, of the positive potassium ions. And losing all that positive would reset the membrane of the sarcolemma back to its resting state, and it would be ready to work again. This happens after we break down acetylcholine using acetylcholine esterase at the synaptic cleft. All right, so that is it for looking at an action potential. And there's some more terminology to talk about when we're talking about that motor neuron and the skeletal muscle fibers. But we're going to look at that in the next video. So make sure that you review this topic if you're a little confused, because you want to make sure that not only do you understand the neuromuscular junction, excitation contraction coupling, and the cross-bridge cycle, but all the details that occur around excitation contraction coupling when it comes to the generation and propagation of that action potential in the skeletal muscle fiber cell membrane, or the sarcolemma.